Hello and welcome to Bio40 Pre-Lecture 15. Today we're going to discuss how mutations were used to dissect the structure of genes. We've shown that genes are on chromosomes, and the key part of chromosomes are the DNA. But is a gene a linear series of base pairs, or is the information spread out along the chromosome? Today we're going to talk about two types of mutational analysis that allowed us to answer this question. Complementation testing allows us to see if two mutations are in a single gene or different genes. And fine structure mapping allowed Seymour Benzer to map where all the mutations in a single gene can be found along the chromosome. We've already discussed complementation in other contexts. When two mutant white flowers are crossed, the offspring can be the wild-type purple color as long as the parents have mutations in different genes. Though one important thing to note is that this only works if the mutant phenotype is caused by recessive mutations. If the mutations were dominant, the heterozygous offspring would still have the mutant phenotype. We could imagine the same system with Drosophila eye color. Let's say that eye color is controlled by two genes, G and R, and we have two mutant flies with mutations M1 and M2. We don't know if M1 and M2 are mutations in the same gene or different ones, so we can cross the mutants and look at the offspring. If M1 is in G and M2 is in R, then their offspring will be wild type because they will still have one functional G and one functional R gene. But if both M1 and M2 are in gene G, the offspring will be mutants because neither parent will contribute a functional G gene. M1 and M2 don't necessarily need to be in the same place within the gene, since multiple mutations can disrupt the same gene. Again, this only works for recessive mutations, because the offspring would still be mutants with one dominant mutant allele and one recessive wild-type one. This is not just a hypothetical scenario. There are indeed many Drosophila eye color mutants, just a few of which are shown here. But are the mutations just different alleles of the same gene, or are they in different genes? Well, we can find out by crossing them all pairwise and seeing which produce wild-type offspring and which don't. The results of such crosses are typically shown in complementation tables like this one. A plus sign indicates the offspring are wild-type, and the mutations are in different genes, while a minus means the offspring are mutants, so the mutations are in the same gene. Let's look at the top row. Of course, crossing two white flies just yields more mutants, but mating a white fly with a garnet, ruby, vermilion, or carnation fly gives wild-type offspring. So the mutations that give rise to those four phenotypes are not in the white gene. However, cherry, coral, apricot, and buff flies do not complement with white flies. We say that they are all in the same complementation group. They are all alleles of the same gene. Since normally only half of a complementation table is filled out, it can be hard to read some of the other genes. The trick is to look in an L shape. Let's see which mutants complement with vermilion. We go to the vermilion column, move down until you get to the vermilion-vermilion cross, then move across the row. We can see that it complements with every other mutation. It's in a different gene from any of the others. Garnet, ruby, and carnation are also in different complementation groups, and thus different genes. In total, that makes 5 different genes controlling eye color. While complementation can help us see if mutations are in the same gene or different ones, recombination can tell us where mutations are located within a gene. Let's say we have two mutants with mutations in the same gene. We'll call them M1 and M2 again. When we cross them, the offspring will have one of each mutant allele and still show the mutant phenotype. But as long as M1 and M2 are in different positions, there will sometimes be a recombination event between the two mutations, leading to some gametes with a wild-type gene and some with two mutations. The problem with carrying out this approach is that mutations within a gene are very close together. If we look at the 5 mutations in the Drosophila white gene, the most distant pair are only 0.011 map units apart, so only 0.011% of the offspring would be recombinant, and only half those recombinant offspring would have the wild-type chromosome. We'd have to score tens of thousands of flies to see a single wild-type recombinant. Clearly, flies are not the ideal model organism for this sort of test. In the 1950s and 60s, Seymour Benzer performed a major series of experiments using T4 bacteriophage, isolating more than 1,600 point mutations in 2 genes. He used complementation to determine which gene each mutation was in, and recombination to map the specific location of each mutation. The T4 phage Benzer worked with are similar to the T2 phage we mentioned earlier in the course, infecting E. coli and getting the bacterium to do the work of making new phage copies. The plate on the bottom left is covered with E. coli. When a phage is added to the plate, it infects a single cell. After the bacteria makes enough new phage, the cell is destroyed, or lysed, and the progeny phage infect the neighboring cells, starting the cycle all over again. After several rounds of this, when enough cells are killed, you see spots of dead bacteria, called plaques. They sort of look like the negatives of colonies, and these plaques have different phenotypes that we can use. The characteristics of the plaques depend on both the phage and the strain of E. coli. Wild-type phage forms small plaques with fuzzy edges in both E. coli B and K12 lambda strains. But phage with mutations in an R2 gene form large plaques with distinct edges in E. coli B and can't form plaques at all in K12 lambda. The R2- phage are called rapid lysis. The larger plaques are the result of the inability to delay lysis. Here's another view of the difference between the large distinct and small fuzzy plaques that the different phage form on E. coli strain B. This difference allows us to perform a screen. A mix of wild-type and mutant phage can be plated, and each type can be identified by the plaque phenotype. The inability of R2 mutants to grow on K12 lambda at all allows for an even more powerful technique called a selection. If you plate both wild-type and mutant phage, only the wild-type will be able to form plaques. If you set up conditions such that the wild-type phage are rare, this allows you to plate the phage at a high density. There are two R2 genes, R2A and R2B, and we can use complementation tests to determine if two mutants have mutations in the same gene or different ones. Of course, this will be a little different than dealing with flowers or fruit flies. We infect E. coli K12 lambda with a large number of both mutant phage. When the ratio of phage to E. coli is large, we say there's a high multiplicity of infection, or MOI. So when both mutants infect a single cell, if the mutations are in the same gene, there still won't be a functional copy of that gene, so no plaques will form. But if the mutations are in different genes, there's now a functional copy of both R2A and R2B in the cell. The phage will reproduce, the cell will lyse, and plaques will form. When the two mutations are in different alleles, we call this the trans configuration. We also do a control with the mutants in the cis configuration, both on the same chromosome. The double mutants are co-infected with wild-type phage. As long as the mutations are recessive, there should be cell lysis. But if either mutation is dominant, it will stop the wild-type phage from functioning normally. It's important to do this control because, again, complementation only works with recessive mutants. Complementation groups identified with these cis-trans tests were dubbed cistrons, which is now synonymous with genes. So to summarize complementation for T4 bacteriophage R2 mutants, the purpose is to determine if two mutations are in the same or different genes. You infect the non-permissive host, that is, the E. coli strain that will not allow the mutant phage to reproduce, at a high multiplicity of infection. We then ask if the co-infection was productive, which is to say, were the two mutant strains together able to produce progeny, lyse the host cell, and form plaques, which would indicate mutations on different genes. Now let's think about recombination in phage. Rather than having a maternal and paternal chromosome as we would in flies, we have two phage chromosomes from different mutants that have infected the same host. But again, as long as the mutations aren't at the same base pair, recombination can occur at a spot between the mutations, yielding a wild-type gene and a gene with two mutations. So to generate recombinants, we infect the permissive E. coli B host with both mutants at a high MOI, allow for one round of phage growth, and then collect the progeny. If recombination is possible, it'll happen at this step. Next, infect the non-permissive K12 lambda host with a progeny at a low MOI. This time, we only want a given E. coli cell to be infected with a single phage, if at all. This is all we need to do to calculate a map distance. Each plaque indicates a recombinant wild-type phage, though it's important to note that the wild-type phage are only half of the recombinant progeny, and the recombinant double mutants are indistinguishable from the parental single mutants. So we take 2 times the wild-type progeny as our recombinant count, divide by the total number of progeny phage plated, and multiply by 100 to get the map distance. A key control in recombination assays is to go through the above process with one mutant phage at a time. Infecting the permissive host with a single mutant, collecting the offspring, and then infecting the non-permissive host does not allow any opportunity for recombination. But sometimes, a spontaneous mutation could cause a mutant to revert to wild type. This control tells us how often this happens. Presumably, the same proportion of phage reverted to wild type in our recombination assay, and we need to make sure not to include them in our count of recombinant progeny. Now, complementation and recombination are very easily confused, so let's take a moment to review how they differ. In complementation, there is no exchange of DNA. The wild-type phenotype appears because each chromosome produces a different functional gene product, together giving the cell both required proteins. In recombination, true wild-type chromosomes are formed due to crossing over. Complementation answers whether two mutations are in the same or different genes, while recombination tells you if the mutations are in the same/overlapping or different positions within the gene. And if they are in different places, it'll help you map the relative positions. To perform a complementation test, you infect the non-permissive host at a high MOI and look directly for lysis/plaque formation, which is indicative of a successful complementation. For recombination assays, you begin by co-infecting the permissive host at a high MOI, allow one round of phage growth to give recombination a chance to occur, and assay the progeny phage by plating on the non-permissive host at low MOI. Here, any plaque formation indicates a wild-type recombinant. I want to clarify what I said a moment ago about recombination and overlapping mutations. Normally we've been depicting our mutations as point mutations, which alter just a single base. But recall that there are also deletions, where several bases are removed at once. In recombination assays, deletions can be identified as mutations that fail to form wild-type recombinants with two or more mutants that do recombine with each other. So take the three mutations shown here. The top two are point mutations in different positions. At some frequency, there will be a crossover between them and you'll see wild-type recombinants. But the bottom gene has a deletion. The thin black line indicates the range of bases that have been deleted. This gene cannot form wild-type recombinants with either of the above point mutations. The base that's mutated in the top gene is missing in the deletion. The correct genetic information cannot be found in either gene, so a wild-type recombinant is impossible. So deletions cannot form wild-type recombinants with point mutations if the mutations fall within the range of the deletion. Two deletions that overlap also cannot form wild-type recombinants. But deletions can still form wild-type recombinants with point mutations outside the range of the deletion, or with other deletions that don't overlap the first. We'll talk in class about how deletions are important tools for fine structure mapping. But for now, let's just take a look at Benzer's results. This is a fine structure map he made of the R2 genes. Each box represents a different one of the over 1,600 point mutations he isolated. There are two important things to notice. First, all the R2A mutations are in one linear stretch of DNA, followed by the R2B mutations in another linear stretch. This is strong evidence that a gene is a linear series of bases along the chromosome. Also note that mutations occur much more frequently at some positions than others. This demonstrated the existence of mutational hotspots, which we mentioned earlier. That wraps up our discussion on complementation and recombination. Next time, we continue to explore gene structure and function with the one gene, one enzyme hypothesis.